Hi everyone, my name is Olivia Reynolds. I'm a PhD candidate at Washington State University, and today I'd like to talk about a short study that we did recently in our lab to identify the effects of chondroitin sulfate and gallic acid on cartilage production by inflamed chondrocytes and alginate hydrogels. To begin this presentation, I'd like to give a little bit of background about osteoarthritis and why it's important that we find new ways to treat this disease. So I'm sure a lot of you listening know someone who suffers from osteoarthritis, or perhaps you have osteoarthritis yourself. Osteoarthritis is a degenerative disease of the cartilage that lines our moving joints. So joints like our knees, hips, shoulders, and elbows can all have osteoarthritis. And what happens during osteoarthritis, as you can see in this picture, is that the cartilage, which is the blue and purple that's lining the ends of our bones, starts to degrade. And as it starts to degrade, it loses its ability to bear loads in the joints, and it loses its ability to reduce friction between our bones. This causes a lot of pain and swelling in patients with osteoarthritis and limits their ability to perform their daily activities. Osteoarthritis is actually the most common joint disease in the United States. It affects over 20 million Americans currently, and that number is expected to continue growing. Because over 80% of patients with osteoarthritis have some form of movement limitation, this is also a top 10 cause of disability worldwide. In addition to causing disability, osteoarthritis is actually a very expensive disease to treat. It costs approximately $40 billion in hospital expenditures annually to treat this long-term disease. So osteoarthritis wasn't traditionally considered an inflammatory disease, but recent research has shown that inflammation also actually plays a large role in the progression of osteoarthritis. So when we injure our joints, that causes inflammatory cytokines such as TNF-alpha and interleukin-1 beta to be activated in our synovial fluid, and those inflammatory cytokines make their way into our cartilage tissue and cause a cascade of inflammatory pathways to be activated. These inflammatory pathways result in an upregulation of catabolic enzymes, which degrade some of the proteins that surround the cells in our cartilage. These include aggregan and collagen 2, and those are the proteins that are really responsible for giving the cartilage its mechanical strength and its ability to bear loads and reduce compression. So when these catabolic enzymes start degrading these ECM proteins, our cartilage gradually loses its mechanical strength, and the ECM continues getting destroyed until our cartilage is completely unusable and completely unable to bear loads. One of the unique things about cartilage is that it also has a very limited ability to self-heal. Articular cartilage has no nerves and no blood vessels, so once it's injured, it's not able to heal itself, and it's not able to produce new extracellular matrix proteins to reduce those proteins that have been degraded. So there's a need for new treatment options for osteoarthritis that restore native cartilage and its load-bearing ability in joints of patients with this disease. So based on that, there's been a lot of recent research in using tissue engineering strategies to produce new healthy cartilage that's able to bear loads and act like native cartilage in patients with osteoarthritis. One of the cell types that's been studied heavily for use in tissue engineering applications is chondrocytes. These are the cells that are natively found in articular cartilage, but they do have several issues that make them difficult to use for tissue engineering. First, chondrocytes are found in very low numbers in native articular cartilage. So in order to use them for tissue engineering applications, you either have to harvest a very large tissue sample, or you have to expand these cells in a lab setting. Chondrocytes, when they're expanded in traditional monolayer culture, tend to dedifferentiate, which is a problem for tissue engineering. These cells lose their ability to produce collagen 2 and aggregan, which are proteins that are found in native cartilage, and they instead start producing collagen 1, which is a fibrous collagen that doesn't have the same mechanical strength that's provided by collagen 2. These cells also become flattened, and they generally just lose their cartilage-specific phenotype as their passage several times in vitro. Third, diseased chondrocytes that are taken from osteoarthritic joints are also inherently inflamed, so we have to find ways in an in vitro setting to reduce the inflammation in those cells so that they're producing healthy ECM around them. So in our lab, we focus on using 3D tissue engineering strategies along with anti-inflammatory compounds to reduce native-like cartilage that has the same mechanical strength as articular cartilage that's healthy. So as anti-inflammatory compounds, we've chosen to use nutraceutical compounds in our lab and study their effects on producing healthy cartilage in chondrocyte cultures. Nutraceuticals are naturally occurring anti-inflammatory compounds that you can find in the food and drink that you enjoy on a regular basis. These counteract inflammation in a couple of different ways. They directly participate in free radical scavenging reactions. So they will react with free radical oxygen species that are produced as a result of inflammation, and they'll scavenge those so that they can no longer promote inflammatory cascades. Some nutraceuticals, such as gallic acid, can also directly inhibit steps in inflammatory pathways, such as the NF kappa B pathway. Gallic acid itself interacts with the NF kappa B dimer protein and inhibits its ability to be transduced into the cell, which stops its ability to upregulate inflammatory mRNA. So when we were looking at nutraceuticals initially, we chose quite a few nutraceuticals and we wanted to evaluate how well these nutraceuticals did at scavenging radical oxygen species. So we ran the ferric reducing antioxidant power assay, or FRAP assay, with several nutraceuticals, which measures a nutraceutical's ability to reduce iron. And we found that gallic acid, as shown by its higher average absorbance on this graph, was able to most effectively reduce iron, which tells us that it also most effectively would scavenge and reduce free radical oxygen species. So we chose gallic acid for further study in our lab to look at how nutraceuticals impact the production of cartilage by diseased chondrocytes. I'd like to talk about my experimental design for a preliminary study next. So the research question for this study was whether the addition of chondroitin sulfate, which is a glycosaminoglycan or GAG that's found in native cartilage, and gallic acid can improve the production of cartilage-specific proteins, collagen, and sulfated GAGs by diseased chondrocytes and alginate hydrogels. So we had 3 groups in this study. The first, we had a group with a base hydrogel material of 3% alginate. We chose alginate as a base hydrogel material because it's been widely used to culture chondrocytes, and previous studies have shown that it can support chondrocyte culture long-term. Our second group had 3% alginate, and we added 1% chondroitin sulfate to the gels as well. Our third group had 3% alginate, 1% chondroitin sulfate, and we added 20 micromolar gallic acid to the culture media. I'd next like to go through the experimental protocol that we used for this experiment. The first thing that we did was we prepared the gel solutions for the experiment. So we prepared an alginate, 3% alginate solution, as well as a 3% alginate plus 1% chondroitin sulfate solution. Next, we added cells to our hydrogel solution. We used passage 2 chondrocytes that were taken during joint replacements from osteoarthritic males between the ages of 60 and 70 years old. We seeded cells at a density of 4 million cells per milliliter in our hydrogel solution. Next, we added the gel suspension to cylindrical molds that had a volume of 30 microliters, and we cross-linked the gels for 20 minutes at 37 degrees in 100 millimolar calcium chloride. We removed the gels from the mold, and then we incubated the cells with 5 nanograms per milliliter interleukin-1 beta to promote the upregulation of inflammatory markers. After a 24-hour incubation with IL-1 beta, we measured the production of nitric oxide via the grease assay to determine how inflamed these cells initially were. Next, we cultured all the gels for 7 days in a standard well plate. And after the 7-day culture, we dissociated the alginate with sodium citrate. We measured the DNA, collagen, and glycosaminoglycan content of our hydrogels. I'd like to talk about a few results from this study as the next portion of this presentation. First, I'd like to talk about the nitric oxide concentration, which was an indicator of inflammation in these hydrogels. So again, we measured nitric oxide concentration via the grease assay, which is a colorimetric assay, and we measured it directly after incubation with interleukin-1 beta, where the cells were most inflamed, and again on the final day of culture. So we found from this grease assay that all of the groups had initial inflammation that was approximately equal based on the nitric oxide concentration. We had a concentration of 4.1 micromolar nitric oxide in all 3 of our groups. When we measured nitric oxide at the end of the culture, we found that nitric oxide was actually undetectable in all 3 of these groups. So from those results, we found that the production of nitric oxide as an indicator of inflammation was reduced equally well in all 3 groups, regardless of whether we added gallic acid to the media or not. Next, I'd like to talk about the glycosaminoglycan content in our 3 test groups. So we measure glycosaminoglycan content with the BlyScan assay, which measures the amount of sulfated GAGs in the hydrogel. We normalize this to DNA content so that we can have an accurate representation of how much sulfated GAGs these cells were producing per cell or per nanogram of DNA. So again, we found that there wasn't any statistical difference between the glycosaminoglycan content among our 3 groups after a 7-day culture period. So we concluded that the addition of chondroitin sulfate and gallic acid did not upregulate the production of sulfated GAGs by these inflamed chondrocytes. Finally, we measured the total collagen content in our hydrogels as well. We did this with the SEER-CoL assay, which again measures the weight of total collagen protein per hydrogel, and we also normalized this to DNA. And for total collagen content, we actually did find a statistically significant difference between the alginate group and the alginate group with chondroitin sulfate and gallic acid. We found that the production of chondroitin sulfate in both the 2nd and 3rd group did increase the total collagen content in our gels, and it significantly increased it when it was added with gallic acid. And so we're concluding from these preliminary results that the combination of chondroitin sulfate and gallic acid may have the ability to upregulate collagen content in chondrocytes. So in conclusion, we found that as far as inflammation is concerned, culture in 3D alginate pellets alone was able to reduce the nitric oxide concentration in cells with or without gallic acid added to the media. Second, we found inconclusive evidence on whether chondroitin sulfate or gallic acid were able to actually upregulate the production of cartilage-specific matrix proteins. We found that total collagen did significantly increase with the combination of those 2 compounds. However, it's unclear whether that collagen was collagen type 1 or collagen type 2. We're not really sure whether that was cartilage-specific collagen or not. And we found that there was no effect on the sulfated GAG content with the addition of chondroitin sulfate or gallic acid. So further analysis is needed in this study to understand if there's truly a benefit to adding these 2 compounds to alginate hydrogels that contain diseased chondrocytes. So I'd like to do some future work with this study. The first is that I'd like to do some temporal mRNA analysis of chondrogenic and inflammatory markers for each gel type. This will help us further understand whether the gallic acid is reducing the inflammatory pathways in the cells. Instead of just measuring nitric oxide, we could have a measure of those catabolic enzymes that degrade the matrix at an mRNA level to better understand what's happening in those cells. Second, we can understand whether the collagen and GAGs that are being produced by these cells are chondrogenic in nature or whether they're type 1 collagen, which we don't want in tissue engineered cartilage. Second, I'd like to culture these cells over a longer culture period to analyze the long-term phenotype of these chondrocytes and the phenotype maintenance in these alginate gels. We only used a 7-day culture period for the studies, which is typically very short for cartilage tissue engineering studies. Chondrocytes are slow growing, so you generally want to give them a longer period of time to mature in hydrogels before you determine whether compounds that you're adding have an effect on the phenotype of the cells. Third, we'd like to add mechanical forces such as shear stress and oscillating hydrostatic pressure to these gels to determine if they have a synergistic effect with these chemical additives that we're putting in the hydrogels. We've determined in our lab in the past that shear and oscillating hydrostatic pressure do significantly upregulate the production of chondrogenic matrix proteins. So we'd like to combine those mechanical forces with chemical stimuli to see if we can further improve the production of cartilage-specific collagen and glycosaminoglycans. With that, I'd like to thank the NSF for supporting this study. I'd like to thank everyone for listening, and I'd be happy to answer any questions that you have during the live session. Thank you.